Hey, hello everyone. Good evening. Welcome. Hope all of
Good
you are
evening.
able to see my screen. Good evening. Hope all of you are able to see my screen and hope all of you can hear me. Can I have a quick
Yes.
confirmation? Perfect. Thank you. All right. Just give me a moment. I don't see. Okay. All of them are from yesterday's session. Anyone? Any new joiners today, or all from yesterday's class? Can I have?
Yeah, myself, my name Maran. Yesterday I missed your this session.
Okay.
Today I have joined, but through the video I have seen your session. Up
Perfect.
to
That's
50 minutes I
yeah.
have closed. Yeah, fine.
Okay, fine. Anyways, yesterday's session was just a very basic introduction to the clinical trials. We haven't entered into uh, anything that need to be taken very seriously, right? We just started, a first baby step, I can say. All right. So today, what we do is I'll have a quick recap about what we have done yesterday, maybe 5 to 10 minutes, and then we go with elaborative clinical trial, and then we set up the base for what we are going to learn for the next 30 days. That is next 30 classes. All right. So let's start with what we have done yesterday. First of all, we all know that we are talking about clinical SAS, right? Again, when I say clinical SAS, it is more of domain. That is, we need to know how the clinical trials work. And then we need to understand where SAS fit into this particular one and how SAS is solving the complex scenarios and how SAS is providing the resolutions to the given problems or given problem scenarios. This is what we need to understand. This is what we are going to do for the next particles, right? So it is a right mix and match of the domain plus the technology. So we started with the clinical trials. We understand that clinical trials is all about from the time we start developing the drug. We are going to develop a drug, correct? We means the pharmaceutical industries. So the moment we start developing drug to till it got approval from your FDA until it reaches the market and reaches everyone. Till that particular one, what exactly happening in the background we are going to discuss. Correct? Yesterday I have given few details. So what we have started, we said that the drug that we are talking here is nothing but it can be a medical drug which can be used to treat the people, right? It can be a preventive drug kind of a vaccine, or it can be a medical equipment. Any of these will fall under the new developments. Correct? What are them? I said the first thing is going to be the drug which can be used for the treatment, any disease treatment, or the drug that we are going to use as a preventive measure, or it can be a medical equipment or medical device. Simply, I can say it. Any of this without approval or without going through a process, it will not come to the market. And that specific process we are calling as a clinical trial. And this clinical trial, if I put it in a proper way, it has certain steps. Let me tell you what are the steps are. So we haven't discussed this in detail yesterday, but we are going to start with this particular thing now. So the entire clinical trials starts with your sponsor. Remember, these terminologies are very, 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 very important. So that's the reason you guys need to concentrate more on understanding this particular terminology, right? So first we are going to have a sponsor. The sponsor is the one who is going to fund your project or who is going to provide all the funding which is required to develop the drug, right? Generally, these sponsors are none other than your pharmaceutical companies. All, or not all, I can say the people who are into the research and development, all the pharma companies are nothing but your sponsors. So your drug development ideally starts with the pharma. So here is what the pharma guys comes to the picture. So these are the drug companies. These are nothing but the sponsors. So the sponsor is the guy who is going to provide the funding for you. Once the funds are available, then we are going to enter into the second phase, or we are going to enter into a facility, and that facility is none other than a research and development facility. Correct? So most of the organizations are going to have this particular one. We can call it as a research and development facility. This is the facility where your drug is actually going to develop, right? But there are few exclusions as well. It is need not to be your R&D department all the time. It can be some other things as well. When I say some other things, those some other things are nothing but it can be of your uh, university, the medical university. It can be of your a normal community clinic, or it can be under the supervisor, or I can say that the doctor itself, the premium doctor itself, even under the doctor supervision also it can happen. But majority of the times, the drug development will happen under the R&D facility of the bigger company, I can say that, or a well-known company, I can say. All right. So let's say that the drug is developed. So they have developed some drug. So what's the next step? This, whatever the drug they have developed, they need to get tested. Without a proper testing, without ensuring the safety and efficacy of the particular drug, it cannot come to the market. So here the clinical trials are going to start. So up to this is only the a smaller picture, correct? We haven't actually entered into the clinical trials yet. Here we got the sponsor who is ready to supply the funds. We have the R&D facility. The guys are really good in doing the research and development, and they are able to develop a drug now. But the drug need to be tested. There are hundreds of drugs will be developed on day-to-day basis, but all these drugs will not come to the market. If someone asks me what is going to be the general range, or what is going to be the span where this particular drug is developed, it is generally 2 to 8 years. In general, I'm talking about. There are drugs which will take more than that as well, right? If we consider COVID, for COVID, we were able to develop the drug within the span of 1 to 2 years, correct? There are few drugs that say that if you consider AIDS, it is, we all aware of this particular thing from quite a long time, but still there is no proper drug for it. Correct? There is no proper treatment for it. There is continuous development, there is continuous research happening around that. Correct? So the minimum years we generally consider as 2, the maximum we can consider as 8, but there are some exclusions as well. Those exclusions are nothing but outliers, right? Those are not fitting to your regular span. But in general, it will take 2 to 8 years. Okay. Now the drug is developed. So drug is ready. So now we will enter into the clinical trials. The very first clinical trial, or the initial clinical trial, will not happen on the human beings. Instead, it will happen only on the animals. All right. We just have developed the drug, and from the drug, we are entering into the very first step of the clinical trials, and the very first of clinical trials is nothing but preclinical trials. Generally, we can call it as a preclinical trials. The preclinical trials is generally carried out on the animals, right? It is purely going to conduct only on the animals, right? The reason why we do this is we want to understand the mechanism of the drug. We want to understand whether the drug is actually working on this or not. Number three, main thing, we want to see the safety. We want to see the safety. By considering these three factors, we generally carry out our clinical trials on the animals. The major outcome that we are expecting here is nothing but the major outcome is safety. Once we understand that, yes, whatever the drug that we have developed is delivering the results on the animals. At least it is getting responded. It is not giving much of the side effects. Then we will enter into the main phase, and that main phase is nothing but the clinical trials phase, and it has 4 different steps in that. Remember, so the clinical trials part has 4 different steps. First one, you can call it as phase 1. Once the phase 1 is successful, we will enter into phase 2. Even phase 2 is successful, then we will move ahead to the phase 3. Once we decide that phase 3 also successful, I'll tell you what and all various things we are going to discuss here, or various things we are going to check here. Once phase 3 also successful, finally we will enter into phase 4. So totally the cycle is going to be phase 1, phase 2, phase 3, and phase 4. Yesterday also we had a little discussion on this, but we haven't go in detail. Today we are going in detail discussion about what we are going to have in each phase, what are the various subjects that we are going to consider for each of this particular phase. Remember, in the phase 1, do you have any idea, guys, who participates under the phase 1? Since we discussed this yesterday, can someone please tell me?
Healthy volunteers.
Healthy volunteers. Perfect. So it generally carried only on a smaller group. We generally say that 30 to 80 people. And remember, we consider only healthy subjects. Remember, hereafter, we don't use the term called person, or we don't use the term called their patients. Instead, wherever we are going to refer someone who is part of this particular clinical trial, we call them as a subject. So we try to adopt the terminology. Remember, we are going to consider only the healthy people, nothing but healthy subjects, right? So the main interest here is going to be on a healthy people. You're going to test your drug, and you're primarily look at the safety. You don't look at the efficiency, you don't look at any other thing. The only thing which I'm bothering about here is nothing but, is my drug safe enough or not? Once you get to a conclusion that the drug is 100% safe, then you will enter into the second phase, the phase 2. The phase 2 is, any idea who and all we are going to test under phase 2? Anyone?
Here it starts with patients.
Wonderful.
Contractors.
Contractors. Okay. Here also, these healthy subjects also can be contractors. Correct? There may not be volunteers all the time. They can be contractors as well. So what else? Only patients and contractors? So can I say that contractors, some of these contractors also patients? Can I consider that? Or how do you want to proceed with this?
You can consider that.
Perfect. Okay, let me tell you. So here we are going to take 3 different types of subjects. Number 1, of course, it is going to be patients, but patients of the same disease. What do you mean by same disease? Anyone? I have developed this particular drug to treat a specific disease, correct? The drug has been developed to treat a specific disease, right? Let's say the disease that I'm talking about here is going to be something called, maybe just consider as a fever. Fever is the disease. To treat the fever, we have developed a drug, right? So now my first set of patients are going to be the people or the patients who are already suffering from fever, who are already having high temperatures, right? The people are the patients with or patients suffering with the same disease, suffering with the disease. What disease it is? The disease to which we have developed this particular drug. That is the first set of people. Number two, the second set of people are going to be again patients, but suffering with a different disease. They are not fall under this particular one, right? Suffering with a different disease. But you can ask me, Murli, can I go with the same? What do you mean by different disease? They may not be, suffering with a fever. Let's say that they have the body ache, right? They are having some allergy. They are having cold. They are having cough. A similar, I cannot say that exactly the same disease or exactly the opposite disease, but the diseases which I can consider as a subclassification are the diseases which have the same prawn as your current disease. All those patients also I'm going to consider here, right? Meaning this one, the second set of people have high chances of getting affected with this particular disease, or the second set of people are maybe because of the tolerance levels or maybe because of their immunity levels, they might not get the fever yet, but they have all the chances to get the fever, the related one. That also we are going to consider here. But three, we also consider healthy subjects. So we have considered healthy subjects in the first one, right? So the same healthy subjects we are going to use here. We are going to test the drug on the healthy subjects, patients suffering with the same disease, patients suffering with a different disease. All these three, the cluster, we are going to consider for phase 2. And the phase 2, we are going to increase the number of people, that is the group size we are going to increase. It can be any number from 100 to 500 people. Some books will refer different number, but altogether it is fine. Either some books refer as 100 to 300, some books refer as 100 to 500, but that is okay. We can consider that it is lesser than 1,000. Anything more than this 80, I can say 100. Anything lesser than 1,000 is a comfortable number. Now, what is the focus here then? The primary focus here is all about safety, right? But safety is not a major thing here. I can say that the focus is on efficacy. How efficient the given drug to treat the given disease. Is it efficient or not? Second, if I say efficiency, the efficiency I'm going to measure on two different things. Number one, how much time it is taking, that is the span it is taking to get treated. How many doses that I need to provide? Can I say that I need to take this medicine every 6 hours, or I need to take this medicine 3 times a day, 2 times a day, or only at night time? How I supposed to give the dosage to get the better? So the efficacy is the primary thing. Along with that, I also check the side effects. Is this drug, because of this particular drug, because I'm looking at a different cluster here, there are group of people, different set of people there. So I also need to check about whether this particular drug is having any side effects, right? While it is treating the given disease, is it causing anything else? Let's say that, is there any itching, or is it causing something called dryness in your eyes, or is it causing dizziness? If it is not giving that particular thing to the people who are suffering from fever, but when I test this particular drug, or when I give this particular drug to the people who are suffering from the body ache and all, so how they are responding? Is the particular drug is actually working on them or not? If it doesn't have any effect, we can say that we know we need to give this particular drug only to the fever, but not to anything else. I'll tell you one of the best example. We all know Dolo, right? The most, I can say that the tablet which is consumed by the most of the people in India during this particular COVID time, right? It has been used use it to treat your COVID. It has been used to treat the fever. It can be used to treat your body pains. It can be used to treat your whatnot. Correct? A simple paracetamol with 650 mg of the dosage, people are using for anything. And there are some memes saying that it is equal to your polo. Correct? So, is the drug that has been developed is targeted only to that particular disease, or it is having the capacity to also deal with a different set of diseases or not? That also will be tested under this. You can ask me now, only why we are testing this on healthy subjects? Why? What is the need? Anyone? Can someone answer this? Because we have already did on the phase 1 on the healthy subjects, right? But here again, why do you want to do this on the healthy subjects again? Why? Anyone?
For detecting side effects.
Exactly. Even Mohanty, yes, you're right. Dosage adjustment. Both of them. How much dosage? Because if you see that you are in the early stage, let's say that the fever level, the temperatures are going to be very less. Let's say that it is somewhere around 100 to 102, right? What might be the dosage? That is again falls under the patients with the suffering with the same disease. There I can use this for adjusting the dosage. But when it comes to the healthy subjects, this primarily, let's say that this particular guy doesn't have a fever. He's not suffering from fever at all. But what happened if this guy takes this drug as a preventive measure, right? As a preventive measure, what happened to him or what happened to her? That can be done under the healthy subjects, mainly to check what are the side effects that we are going to get if I go with the healthy subjects. All right, so that is about your phase 2. Then when I enter into the phase 3, here this is the most crucial thing. I can say that even though the marketing and everything will happen in the phase 4, but phase 3 is the most crucial in the clinical trials. Why it is crucial? Because we are expanding our base drastically, anywhere from 1,000 to 5,000 people. Here again we are considering a random set of people. Here we are considering the healthy subjects. The same 3 things are going to repeat, but when it comes to the healthy subjects, when it comes to the patients with suffering with the same disease or with a different disease, here we are going to consider a lot of other factors as well. Meaning we are going to consider the gender, because some drugs, even though the disease is going to be the same, for gender it is going to be different, right? So when we are going to have this particular drug, the doctor is going to treat differently for female, and the doctor treatment is going to be different for the male. Even though the disease is going to be the same, there are some specific diseases, correct? Because our body couldn't tolerate few of the drugs, right? So that is the reason we need to consider the various gender people as well. We need to take the mixed group of both male and female, right? Let's say that up to this, we are concentrating only on the male subjects or only on the female subjects, depending upon the type of the drug. But the moment you enter into the phase 3, you're going to test it on both the genders, just to see what happens if I give this particular drug to the other gender. What happens? Because side effects we need to see. Second, some drugs, the dosage we need to decide, because here my focus is on the safety. What happens if I give to the other gender? At the same time, I need to check the efficacy here. At the same time, I also need to check the side effects here. All 3 are major things. If you see the second one, I'm not going to bother much about the safety, because safety is already tested in the phase 1. But here, since I extended my base to the large people, because I'm talking about various genders, we are talking about various age groups. The age groups are also going to vary, because we're going to consider all the age groups, right from your, let's say, from the toddler to till your old age people. And we are going to consider the various continents as well, because based on the continent also these will be changed, right? What not? There are so many other factors we are going to consider. What are those factors? What are those parameters? Again, we are going to discuss in the later part. But on a whole, right now, all we are going to concentrate here is we primarily focus on the various parameters while selecting the people. We also use some of the techniques here called randomization and all, but it is too early to discuss about that. So we'll discuss about the randomization and all. How do we take random people and all later? All right, but the primary focus is on safety, efficacy, as well as the side effects. Once we are through with this particular one, we are ready to launch our particular drug to the market. So here, phase 4 will start. Phase 4 is everything. Diverse subjects, yes, true. Maybe the right technical word, if I say Maithili, yeah, I can say that it is going to be test on diverse subjects. Wonderful. Because we are going to consider everyone here, right? We are going to consider everyone here. All right. Now, the next thing that I'm going to talk is the phase 4 is one step before you enter into the market, just one step. So here we are going to extend our range from 10,000 to 50,000 people. A lot, correct? But here the major thing that you need to understand here is lot of factors come to the picture. A lot of factors, meaning you have given, the sponsor has done lot of funding so far. They have given a lot of money to the R&D facility. They have provided a lot of money to get your contractors, get your volunteers. Sometimes if the volunteers are not there, they need to pick the people from the specific vendor group. She have done a lot of work here. They have put a lot of effort to bring this particular drug. So there is a lot of capital involved there. Correct? According to this particular capital, they need to fix the price. They need to fix the dosage. They need to fix the stock, how much volume. Because if I'm ready to give this particular one, if you remember during the COVID time, even if you take Bharat Pharma or anything, before even we get the approval from the government, right? They have produced, let's say, 1 million stock. I already have 1 million doses with me before even we go with the approval, because once the approval comes, we should be able to provide the minimum number of dosages to the cluster of people. So the capital, the price, the store, the dosage, the stock, whatnot, even the complete the marketing strategy, all my marketing strategies as well will be decided here. This is just one final step before we enter into the market. So here we don't talk about the safety, efficacy, side effects, because it's all done. Here only we talk about the external terms, which are nothing but what is my capital I have invested so far. So what might be the price for my given medicine? What might be the dosage for each of the age group, each of the gender, or each of the all the diverse subjects? Right? How much stock I should have before, at least if I enter into the market, what might be the minimum dosage? Because if a doctor, prescribe this particular medicine to a patient, let's say that for the set of patient, let's say each patient he suggests that you need to take this medicine 3 times a day for a month of time. So one patient itself is going to buy 90. That is nothing but approximately 100 tablets he is going to purchase. Right? So let's say that if I'm giving this to a particular country or to a particular state, I need to release with minimum number of stock. So the stock need to be maintained here. Post that the marketing strategy, you need to approach the right facilities. You need to approach the right doctors to make sure your drug is part of their prescription. So all those marketing strategies also part of your piece number 4. So with this, with the stage number 4, ADRs in target population. True, but Maithili, I'm not going too much into it because I know that you sounds really good with your functional things, but I'm not going that deeper because I'm looking at the launch. Right? Let's use all this terminology a little later. Perfect. Now, once we are done with phase 1, 2, 3, 4, we are ready for this particular drug. But if I want to release this particular drug to the market, I need an approval. Am I correct? Without an approval, I cannot do. So to whom I need to approve, or to whom I need to reach for the approvals? Anyone? Without an approval, I cannot do. So who are going to be the approvers? Can someone please tell
FDA.
me?
FDA.
FDA. Okay.
Drug regulatory authorities.
Drug regulatory
Authorities.
regulatory authorities. Okay.
TCG.
MHRA for Europe.
Someone said DC.
MHRA.
TCG.
Someone mentioned
That is
European countries. EMA.
DCGI. That is
DCG. Sorry, I'm just
CDISC.
typing what you are saying.
I.
Okay, there are a lot of things are happening.
I.
Okay, yeah. We'll go with the lighter note. Let's consider three different markets for now, because most of this started from the USA. So for USA, we have FDA. What is FDA? Food and Drug
Administration.
Administration. For Europe,
MHRA.
MHRA is there. There is another authority as well. We can simply call
EMA.
it as a EMA
MAS.
as well. Yeah. What is EMA? Anyone?
European Medicine.
A for authorities. You can also consider as authorities or agencies. Okay. For India,
Drug Control General of India. DCGI.
DCGI. Correct. So we have drug control governance in India. We have for Europe, we have European medical agencies or medical authorities. For USA, we have Food and Drug Administration. So these are the regulatory authorities, right? So there are different authorities as well. Primarily, we consider these, right? Majorly, most of the times we follow FDA, right? Okay. So if I want to go to these approvals, what I need to do? To get the approvals, I need to prepare some reports, correct? I'll say that this is my report. These are the various subjects that I have considered. These are the dosages that I have provided. These subjects are suffering with these set of diseases, main disease, primary disease, as well as the child diseases of that. And after I use this particular drug, it got treated in these many number of days. That is nothing but a span. After recovery also, what are the various precautions that we need to take? And finally, if any side effects, we need to mention that. Correct? If you enter any drug, if you just, let's say, even if you put Dolo 650 in your Google, if you go to a 1mg site or any site, it is going to talk about what this drug is, what is going to be the composition of this particular drug, why we use this particular drug, what are the side effects. All these four parts will be there, correct? Even the dosage information will be there. So all these are supposed to be part of your report. I'm telling on a very lighter note, right? Generally, we don't follow that, but on a very lighter note, these are going to be the various reports we have. All these reports, we are going to do it in SAS. To prepare this particular report, we are going to use SAS. Where SAS is coming to the picture in the clinical trials means while we produce our reports to the FDA or to the regulating authorities to get the approvals, we need a report. That particular report, we are going to do it in the SAS. Fine. Mostly you're saying that SAS, but SAS is somewhere, it got developed in 1960. We know all the things and all. Let's say before SAS, before using the SAS, are we doing on any other technologies? Yes. Before we standardize this particular process, before FDA standardized this particular process, everyone was using their own way of representing and their own way of producing the results to the FDA. No one is going to follow a specific format, right? For an example, right here I have the approvals part. So before I enter into this particular approvals part, everyone was following a different set of reports. For an example, there is a company one, right? So they have produced one drug. Let's say we can call it as a drug one, and this particular drug one is to treat a specific disease. So they have generated a report, and this report they have submitted to FDA. Similarly, to treat the same disease, there is another company, they also produce the drug. All right. So when they produce the drug, it is going to be drug 2, and they have generated their report on their way. Right. Similarly, drug 4. Now there are 4 different reports for 4 different drugs, but all of them are going to treat the same disease, and FDA has received all these 4 reports. Now, FDA need to go through each of the report, and they need to provide their approvals, or they need to say whether it is approved or rejected. Though, let's say that report 1 is prepared in an Excel, a report 2 is prepared in Word, a report 3 is part of PDF, a report 4 is in Excel. Because FDA have not asked for any specific format, so each company is producing their own report. So the first thing is, because it is difficult, if I want to compare an Excel with Word, or Word with a PDF, or PDF with XML, it is quite difficult, correct? So I need a specific format to compare the results as well, just to see which company is having the better trust, correct? Because the parameters are going to be the same. So what they have done, they first standardized, because I need to have some standardization here. I need to place some standards. So to come up with the standards, because there is a need for coming up with the standards. The standards include the very basic stuff, which is nothing but the format. In which format you need to prepare a report. First thing. Number 2, once the format is fixed, let's say that everyone is sending their report in an Excel, right? So one guy, when they say about the continent, he used a term called continent. The company 2, they use it as a region. So continent here they mentioned as Asia. But when it comes to the second one, they use the same Asia, but they don't call it as a continent. Instead, they use it as they call it as a region, right? There is a third person, they simply call it as an area, but all of them are referring the same thing. Let's say all the 3 people are talking about the continent only. But the first company used the word called continent, that variable name. The second guy used region. The third guy used area. So there need to be some set of standardization when it comes to the parameters, correct? What parameters to be part of your report? Again here, report number 1 has 100 parameters, 100 different things he have considered, and he presented the report. Whereas my the report number 2 will have only 50. Whereas my report number 3 will have only 15 parameters, which are critical to that particular one, right? So FDA also has given the details about every report that you are going to produce to me. The report should have some mandatory parameters. Those should be common for all of the reports. You may have some additional parameters that may not be mandatory to me, but I'm specifying that these are the various parameters you should follow. That is the number. At the same time, what are going to be the parameters? Those are critical parameters, or those are going to be the normal parameters. Both of them also I'm setting up here. I means someone is setting up this. Who is the someone we are going to talk? Number 3, once the parameters also specified, now we are talking about the values. Let's say someone asked to mention country details. I'm typing as India. Someone saying IN. Someone saying IND. All 3 are valid. For me, country is India. For someone, the country they are just using an acronym IN. Some using simply IND. All 3 are possible. All 3 are correct only. But there need to be some set of standardization, right? If someone says that country should be of 3 letters, then you have to use IND everywhere. Someone has to mention that it is going to be 3 letters, then everywhere it is going to be 3 letters only. It should not be elaborative one. It should not be a short one. It's supposed to be 3 letters everywhere. Correct? So standardization should be happen even to the values. Like this, there are a lot of parameters which are required. Mainly those are required to do one thing, that is globalization. Right? I need to globalize. I need to globalize my standards so that whatever the report that I am getting, all the reports, I can quickly scan it, I can quickly do the approvals, or quickly I can do the rejections. All these three I can do because I know the format, I know how to validate. Even if I want to provide some information to the other person who want to validate it, it is going to be quite easy for me because it is under the standards that I have already set up. So to set up this global standards, FDA has come up with one organization, and that organization is nothing but CDISC. Any idea what is called CDISC? Can someone expand this particular acronym? Sorry? Anyone?
The clinical data interchange standards consortium.
Yes, exactly.
Clinical
So
data interchange standards consortium.
exactly. Yes. You guys are correct. CDISC is the main standard organization. This organization is actually funded. It is actually started with the help of your FDA. FDA is the one who started this particular one. The acronym stands for CDISC is mainly is going to provide end-to-end global standards, right? It called as Clinical Data Interchange Standards Consortium, right? So this particular one is started somewhere in the 1998, right? So they are going to provide, it is a very open thing. You need to say that it is a open organization, and anyone who want to know the standards, they can simply visit cdisc.org website, and they can download an implementation guide. That is nothing but it will be call it as SDTM implementation guide. So maybe I need to elaborate few things here. This CDISC, after they finalize all the global standards, for the clinical trials, they have given 3 sets, or I can say that 3 different standards. Number 1, SDTM. Number 2, ADAM. Number 3, TLF. Any idea about this? Anyone? Maybe basic idea about what is called SDTM and what is called your ADAM and what is your TLF? These are the
Study
different.
data tabulation module.
Yeah, you can also call it as, either you can consider it as a study data, or you can also call it as a standard data. Standard, or you can also call it as study. Some books refer standard, some book refers as a study. Standard data tabulation module. ADM stands for
Analytics, analysis data.
analytical or analysis data models. Right? Okay. TLF stands for tables, or
Tables, listings, figures.
listing and figures. So in our course, we are going to start with SDTM, and then we will enter into ADA, and also we are going to cover TLF. All the 3 things we are going to cover in the course. All right. So these 3 can be considered as 3 different standards, or 3 different standards that we are going to follow to generate the report to send it to the FDA approval. Every report that is going to receive by your FDA has to follow this particular one and has to be in the same format. All right. For SDTM, currently we have 3.3 version. There are various versions as well, but most of, I can say that 90% of the pharma industry is still under 3.2. the implementation guide, which can be considered as a Bible. So the standard data tabulation model implementation guide, which is going to be 3.2. We are also going to use 3.2 version. And when it comes to the Adam currently at 1.1 version. For TLF, there is no specific thing, there is no specific version and all, but we are simply go with what are the various tables, listings, figures that are required, what are the supporting SAS procedures for that we are going to discuss. Questions? Any questions on this? Anyone? Right. So the next thing I would like to talk here is, this is another thing, because I'm also going to provide all these notes to you guys, no worries about it. So today is only the initial thing. This particular one doesn't require much of the notes, but anyways, I'll save this particular Excel. And whatever I have discussed so far, the basic details I have already provided here. Maybe I can share this with you guys as well. And also, maybe I'll start with this particular one, a very small topic about how the clinical investigation start, what is going to be the protocol. Remember, irrespective of whatever the clinical trial you talk about, all these clinical trials, all these clinical investigations begins with the protocol. The protocol is nothing but it's a document describes about how the trial is supposed to be conducted, how the clinical trial is supposed to be conducted. You can see that these are the various steps I have mentioned here, right? How these steps need to be started, how we are going to carry out this particular one, how to conduct this particular one, meaning the objective of this particular clinical trial, what is going to be the design, what is going to be the methodology that you want to follow, what are the statistical considerations that you want to do, and who is going to be the organization, which organization is going to start this. That is nothing but the sponsor, or it is going to be called it as the person who is going to fund this. All I'm doing this just to ensure that the safety of the trial subjects, correct? Because you're doing this on the human beings, right? So when you're doing this on the human beings, the first utmost important thing that I have to consider is the safety. How safe this particular clinical trial is going to be. Why you are doing this? What is going to be your design? What is the methodology that you are going to follow? What are the statistical considerations? That is nothing but your measures, right? All these I'm going to consider. Okay. What is that particular protocol document is going to describe? It is going to describe a lot of details. Can you see this title page, background, objective, study? There's a lot of stuff will be there. So our next topic into your clinical trials is going to be how your protocol document is going to be, why you are going with the protocol document, and what is the purpose of it. So before we start entering into your SDTM, that is going to be the entry phase for our clinical SAS. Before we go there, we need to understand few more things. That is nothing but we need to understand about what is the subject is all about, how we consider the subjects, and there are few terms and terminologies that we need to understand. It may take 1 or 2 days. I can say that minimum 2 days, 1 hour classes are required to enter into the SDTM. Once we enter into the SDTM, the story is going to be different. We are all going to talk about the raw data. Then we start discussing about the various data models and all, right? So before we enter into that particular one, we need to understand some clinical terminology. And the clinical terminology starts with your protocol. And this one we are going to discuss in the tomorrow session. So today's session you can consider as an elaborative session on the yesterday's one, because I can see that some of the new joiners as well. At the same time, I want to expand on each of the phase one. That's the reason I have done this here. So from tomorrow's session onwards, we are going to talk about the terminology. The main concentration is on the terminology. So we understand the terminology. Once we are good with the terminology, then we will enter into the SDTM phase. Clear, everyone? Can I have a quick confirmation? Any questions or doubts here? Please let me know. I'm happy to discuss. All right. If there are no other questions, we can wind up the session today here. And tomorrow we have a session, and in tomorrow's session we start discussing about the various terminologies that I can use it to before we step into the SPTM, ADAM, and TL. Clear? These are the initial days, guys, before in a slower pace. Okay? Thanks, everyone. See you tomorrow. Take care.
Thank you.
Thanks, guys.